Hello, in this video, we're going to discuss microbial mechanisms of pathogenesis. So moving into chapter 13, 14, if you hear the term patho is going to be relating to disease. And Genesis, think about like the book of Genesis. It's the beginning or how a disease would arise. So pathogenesis, by definition, is the mechanism a microbe would use to cause disease. So if you hear of pathogenic microbes, those are microbes that cause a disease. Or pathogen is an agent that can cause a disease. And the reason that they're able to do this is because of virulence. Virulence is the ability for a pathogen to infect a host and actually cause disease. It's not related to the severity of the disease, but more than likely the probability or the ability that the pathogen can actually cause the disease. So we're going to talk a lot about virulence and virulence factors here coming up. But when you think about a pathogen, this is an agent that can cause a disease. They have a goal in mind. They have to get inside the host. Not only do they have to get in, they have to adhere on and actually establish themselves. They have to bypass the immune system in order to colonize, and then they can gain access to nutrients and eventually leave the host. So they have lots of things to do. And if you have a healthy immune system, chances are these pathogens would not succeed. However, with being immune compromised or again, those virulence factors, it makes that likeliness they have a lot of a greater chance producing a disease. All right, so thinking about those steps and those goals, they have to adhere to the host cells. So they have what are known as adhesion factors. And in bacteria, they can have potential pili, which are going to be adhesions. And they may even have surface proteins that allow them to bind onto the host receptors. So you may see that they have attachment of proteins, like especially for viruses, or again, you may see like pili, for instance, that would allow for a bacteria to adhere on. They also may have that capsule. And if you remember talking about bacteria having a capsule, they have their cell wall and then they have this covering over them called a glycocalyx. Because they have this capsule, it allows them to adhere on as well. And it makes it a lot more difficult for our white blood cells or our immune system to actually get a hold of them. On top of that, it also allows them to have biofilm production as well. Then they have specific host interactions. And what will happen is those adhesions and those receptors really determine how specific the pathogen can be to a particular disease or a particular host. So, not every pathogen causes every disease. This pathogen is going to have a specific host. And because of that, having a specific host, it can accomplish a particular disease. For example, Neisseria gonorrhea. It has adhesions on its fimbriae, to adhere to the cells of a vagina. And it can also attach to sperm cells, but it doesn't infect other hosts. So it's only going to have those specific receptors. So the pathogen is very, very specific to a particular host, and again, in these particular cells of the host. So down at the bottom, you can see this picture here of those pili for bacteria. And again, it has those adhesions and they can bind on to these receptors on the host cell. And again, this allows them to have that adherence and be able to attach on. Then again, we get into more detail about those virulence factors. So again, virulence is the ability a microbe has to cause a disease. It doesn't do anything with severity of the disease, but it's the probability or the likeliness that they can cause disease. And when we discuss virulence factors, these are traits a pathogen uses to produce toxins such as exotoxins. It also allows them to gain access in nutrients and be able to produce other enzymes as well. When they produce extracellular enzymes, it allows them to actually dissolve structural chemicals in the body, and it allows them to further invade and gain access to nutrients. Like I said before, it allows them to produce toxins, specifically exotoxins. Exotoxins are different than endotoxins. We made mention of endotoxins when we talked about lipopolysaccharides and gram-negative bacteria, but exotoxins are secreted by the pathogen in a healthy cell to destroy the host cell. So sometimes they will have what are known as cytotoxins. These are going to kill the host cell. You can have neurotoxins that are going to interfere with nerve function, Or you can hear of enterotoxins, which would cause damage to the GI lining as well. And they can cause toxins that will cause membrane damage. So it actually disrupts the plasma membrane. And you can also see of hemolysins as well, which are going to destroy blood cells in particular. Now, you can also see endotoxins as well. So be careful with endo versus exotoxins. Exotoxins are secreted in a healthy bacterial cell. They don't have to have damage or anything like that. They just secrete these toxins to basically destroy the host. Endotoxins are going to be found in gram-negative bacteria. When they're found in gram-negative bacteria in that outer membrane as part of their LPS, lipopolysaccharide. It's also known as lipopolysaccharide A or lipid A, and it's released when gram-negative cells die, or if they divide, or if they're digested by phagocytic cells. So if they die for some reason, if they're dividing, or again, if they have been digested, they will release the endotoxin. And this endotoxin can cause what we know as toxic shock. It can cause fever, inflammation, and even diarrhea as well. One really common example that when we discuss endotoxins with LPSA or lipopolysaccharide A is the use of superabsorbent tampons in the 1980s. Women were wearing tampons because the tampon said that it could allow for absorbance for up to 24 hours. And what they realized is actually they were causing women to go into toxic shock. And so that's one of the reasons and why they've changed tampon use as well to say no more than 8 or 10 hours. And again, because of the fact it could cause toxic shock due to these endotoxins. All right, other things about virulence factors, they're a trait a pathogen uses to also escape the immune system. Our immune system is supposed to detect these unwanted antigens, these foreign bodies. But because they have these virulence factors, they can escape that detection or the removal. And they have what are known as anti-phagocytic factors. So phagocytosis is a process by which our white blood cells can actually grab on to the antigen and engulf it and then use these digestive enzymes to break it down. These particular pathogens have what are called anti-phagocytic factors. So this allows them to avoid phagocytosis. So really quickly down at the bottom, and if you follow along in chapter 15, you can see these steps in more detail of how phagocytosis takes place. Again, this is going to be found in our white blood cells. Usually you will talk about neutrophils, we're going to talk about macrophages and dendritic cells in particular for phagocytosis when we get into more detail in chapter 15. But basically what will happen is the phagocyte is moving around, it recognizes this antigen and it attaches to it. Once it realizes and it basically determines that it's foreign, it engulfs it or it ingests it. And when this happens, it's called a phagosome formation. Then finally, what will happen is the phagosome will fuse with lysosomes. Lysosomes are those enzymes that are digestive enzymes. And this allows to have digestion of that antigen. And then finally, once the antigen has been broken down, now it can be released through exocytosis and it's basically rendered useless and it's been killed. So with these virulence factors, they have anti-phagocytic factors. They can avoid the immune system. They can avoid those unwanted pathogens or unwanted phagocytes. And one of these methods that it can do is utilizing those capsules. So as I said before, a capsule is what we think about that glycocalyx. It can be a capsule or it can be a slime layer. But capsules' jobs are to basically make sure that the immune system doesn't recognize that the foreign agent is foreign. Even if it does recognize that it's foreign, it makes it really difficult for our white blood cells to adhere on because the capsules are kind of slippery. And so therefore they can't have that proper receptor recognition format and be able to have that proper adherence. They also have phagocytic chemicals or anti-phagocytic chemicals in which the pathogen can survive even though it's actually undergoing phagocytosis. It prevents that fusion of the lysosomes with the phagocytic vesicles. So even though they've been eaten, basically, they can still withstand and survive that phagocytosis and that digestion occurrence. because they truly don't have that fusion taking place. And again, they're going to avoid our antibodies. Our antibodies have a very specific shape to them. They are a Y-shaped protein, and they are produced by what are called B cells. We're going to talk in chapter 15 about the idea of T cells and B cells, or B lymphocytes and T lymphocytes. B lymphocytes are going to produce plasma cells, which are our antibodies. Antibodies are also known as immunoglobulins. And antibodies are really, in particular, useful for a very specific response called the adaptive immune response. Like I said, we'll talk more about that in chapter 15. But antibodies interact with antigens. Antigen is the foreign agent. So your bacteria would be the antigen. Your antibodies would be yourself that are producing these as a result of these antigens being present. And the antigen-antibody complex, when they come together, are very specific, like a lock and key model. When you look at the antibody as well, that Y-shaped protein has what's known as an Fc stem and what's called a variable region. The Fc stem actually is going to bind with the antigen, and the variable region also attaches to the antigen as well. But what the Fc stem does is it acts as a flag for phagocytes, kind of targeting it and letting you know where they are, so they can be flagged for phagocytosis. And again, thinking about this as well, again, avoiding our antibodies, what can also happen is we have IgA in our body secretions. And what can happen is they have these proteases that help to actually break down our antibodies. So this can happen especially for bacterial adhesions on the pili. They can secrete proteases, these specific enzymes that break down our proteins, which are antibodies. What also can happen too is because they know that there's adherence to those pili, when the bacteria recognize that those pili are basically under attack or they've been bound, they can lose the pili, just like an iguana can lose its tail when it feels threatened. It can actually basically shed or, like, in a weird way, cut off its own tail, and it can grow back a tail. Same thing can happen for bacteria. They have that pili, and when they realize it's sort of been compromised, if you will, they can chop that off and basically make a new pili, so they have a rapid turnover. They also can have antigenic variation where they genetically alter their pili. So again, those antibodies cannot bind specifically. And again, that would cause issues with those Fc receptors and that bounding and flagging. So gonorrhea in particular produces chemicals which prevents that fusion of the lysosome, and it also secretes protease, which break down IgA and mucus. All right, so this just shows you a little bit of that picture. So in purple, you can see the antigen, which is that bacteria. And then the Y-shape figure that you'll see is the antibody. You'll see that there's what's called a Fab region and there's an FC region. The Fab region is what's going to bind onto the the Fab region is what binds onto the antigen. The FC region is going to recognize that it's foreign, and that's going to be sort of the first little marker, if you will, to trigger that step for phagocytosis. So when you're looking over at the bacterium, you're going to see that the Fc receptor on the bacterium binds with that Fc region on the antibody. So when we see those pieces taking place, we can see how these bindings are happening. And again, once the Fc region recognizes that this is going to be something foreign, it can bind that region again to phagocytes and initiate phagocytosis. And then eventually what can happen is this microbe, this pathogen, if you will, can leave and find a new host, and it will leave via those portals of exit that we made mention of previously in chapter 13. All right, so getting into some new topics here, we think about epidemiology. Epidemiology focuses on the effect a pathogen will have in a specific population.